Good day, everyone. Welcome back to my YouTube channel. Our last discussion was about the protocol of this enzymatic assay, where we talked about the lipase agar, the protease agar, the chitinase agar, the xylanase agar. I remember mentioning that we use our gold standard was the lipase because of the similarity between the structure of both lipase and plastics. I remember talking about producing a lipase agar, the lipoidal emulsion, and the base media that was mixed together with OWB agar, and then an acid lift of the colony into the agar. So now this, without the bacteria, the coloration of a lipase agar is pink. That's one, using OWB as substrate. Now I remember mentioning using a fluorescent, an ultraviolet fluorescence of 365 nanometer. You can see the clear zone that was produced by this enzymes. Now this is illuminating because we used the fluorescence to check it. Yes, there was a growth, but it was important to check with the fluorescence because if now there are two things that you will observe in this picture. You can see how the bacteria are glowing. This glow that you see, they are bacteria. You can notice the glow. Sometimes there could be growth, but the bacteria will not glow. When there's no glow from the bacteria, they are not positive for lipase. No. There could be anything. But the moment there's a glow, there are colonies, there are halos, and there's a glow. That makes them positive for lipase. I said that I was going to show what it looks like, and this is a picture. Then further, we also did a protease test. Remember, we talked about using skimmed milk agar. That's what we did. We can notice the halo. So now, if we measure this diameter, that's the millimeter of the halo. That's the growth rate. Measuring this is the growth rate. These are the halo. They are clear zone. This is showing the interaction between the bacteria and the substrate. So they are showing that the bacteria can feed on this substrate, and they will measure the diameter of this clear zone to know how much growth out of there. This is the skimmed milk agar, and showing growth and the halo. Of course, already the presence of an halo already indicates growth in the bacteria. So now we need to understand that those that are positive for lipase are the ones that went further for downstream analysis. Anyone that is not positive for lipase is discarded. And now also, some of them could be positive for lipase and negative for other tests. We just need the one that will give us, we just want to pick the strongest bacteria in all of them. For instance, we have 5 tests. We have the lipase test, we have the protease test, the chitinase test, the xylanase test, and the cellulase test. For a bacteria that is positive for all 5 of them, those are our best bet to use in the biodegradation analysis. The biodegradation analysis is where we pick these bacteria that are positive for this enzyme and introduce plastics to them. We believe that those bacteria that are positive for at least 3 of these enzymes are going to be able to be instrumental in the degradation of plastics. Now, going forward, we also did the cellulase activities, and the cellulase activities demonstrated by the hydrolysis of carboxymethyl cellulase on CMC agar plate. This is the CMC agar plate. You can notice this red coloration. This red coloration is due to the rinsing of this agar with methyl red, so that we can see it is a growth and we can see the halo. So now, rinsing into methyl red shows us that there's the bacterial growth, and we can also notice the halo, the clear zone of inhibition that is caused by hydrolysis. That's for cellulase activity. We also went further to carry out chitinase activities. This is chitin. Yes. You can notice the halo too. You can notice the bacterial growth and the halo. Now, if see also for the chitinase test, you can notice what these particles that are on the particles that are settled in this plate. Now, this plate was also sodium chloride was used to rinse this plate. That's what makes the halo visible. Remember, we already shared the protocols. In protocol, we mentioned all that we used. Now, because chitinase is very heavy. Now, it takes a lot to dissolve. In fact, in the production of the chitinase agar, we had to dissolve chitin in concentrated hydrochloric acid. And that's why we have these little particles there. But you can see that this so we didn't do just one agar lift. The same, we lifted a colony and plated it in three different positions. Wanted to be sure. For this visualization, we noticed how the three bacteria were positive. They grew. They were able to be, after inoculating the bacteria, there was growth. Now, what we're trying to even find out with this is that maybe only one will grow, then it's telling us something. But in this case, the three acid lift that we did were positive and then they grew. So in this case, not a mistake, but that the bacteria could hydrolyze chitinases. Then we went further also to do a xylanate test using xylene. Now, the plates were incubated for 48 hours, then treated with Congo Red for 15 minutes, followed by a sodium chloride wash, revealing the hydrolysis of the xylene. Mira, look at it here. Because actually, the Congo Red is the reason why this plate is red. This is the actual color of xylene. Xylene is a light blue, has a light blue coloration. But the reason why we rinsed with Congo Red, we treated the plate with Congo Red because we want to see if there's an actual growth. You can notice how the Congo Red just single-handedly brought out our growth. Here is our growth, showing the xylene and the bacterial growth. So these are the results that we're going to get when we carry out our experiment in this manner. Thank you for listening. In subsequent videos, I'm going to show us how we now pick these bacteria that are positive for at least 3 of these enzymatic activities and went through the experimental analysis.